How are the functions of the digestive system regulated and controlled? It turns out that the GI tract has its own nervous system that's referred to as the enteric nervous system. It controls movement, that is motility, of food and waste, and it also regulates the secretion of various digestive glands. The yellow bundles in this image to the left are the neurons of the enteric nervous system. So there are two different reflex arcs that control digestive system functions: short reflexes and long reflexes. Short reflexes are mediated by the enteric nervous system and respond to stimuli that are internal to the GI tract. The stimuli that these reflexes are sensitive to are changes in GI stretch, changes in pH, nutrient concentrations, or solute concentrations. So recall that effectors are structures that respond to reflex stimuli. The primary effectors of the digestive system are the smooth muscle bundles in the muscularis mucosa or the muscularis externa, or the various glands that secrete digestive juices and digestive enzymes. The responses of those effectors involve smooth muscle contraction, smooth muscle relaxation, increased secretion from glands, or decreased secretion from glands. The response tends to be specific to the specific type of stimulus that's detected. So on the other hand, the long reflexes are mediated by the central nervous system. These reflexes tend to respond to stimuli outside or even inside the gastrointestinal tract, but primarily they respond to stimuli outside the gastrointestinal tract. For instance, seeing, smelling, tasting, or thinking about food can influence digestive system activity in preparation for receiving a meal. The central nervous system then communicates with the enteric nervous system, and it's the enteric nervous system that stimulates or inhibits the effectors. So again, stimulation or inhibition will depend on the stimuli that are received. Digestive system activity is provoked by mechanical stimuli like stretch of the walls of the alimentary canal, and is also stimulated by chemical stimuli. So for instance, changes in pH or the accumulation of the end products of digestion, such as monosaccharides, amino acids, or fatty acids. So recall that effectors are structures in reflex arcs that carry out the body's response to the stimulus that started the reflex. The primary effectors of digestive system activity are smooth muscle and glands. So like other types of signaling that we have seen in the body, both neurons and hormones control digestive activity. Digestive hormones come from the stomach and from the small intestine, and we'll examine two of them now. So we'll focus on two hormones that are shown in this table here, Cck and Gastrin. So cholecystokinin, or Cck for short, is a hormone that is produced by the mucosa of the duodenum. So recall that the duodenum is the initial segment of the small intestine. And the main stimulus that triggers the production of Cck and its release is the presence of fatty chyme. Chyme is the paste-like food residue that leaves the stomach and enters the duodenum of the small intestine. High concentrations of fat in the chyme trigger the release of Cck, and then Cck targets the liver and the pancreas, which causes the liver to secrete bile into the small intestine, which assists in mechanical breakdown of fats into smaller droplets, and it also stimulates the pancreas to release fat-digesting enzymes into the small intestine. Gastrin is a hormone that's produced by a type of cell called the G cell in the mucosa of the stomach. Its primary stimulus for release is partially digested proteins within the stomach. Gastrin targets the stomach itself, causing increased release of HCl, which is hydrochloric acid, which aids in the chemical digestion of proteins within the stomach. Gastrin also stimulates muscle movements in the small and large intestines to help propel food masses once they have reached those organs.